Good morning, everyone. So today we'll follow the Cambridge syllabus, and we are going to start with chapter 4, which is biological molecule. So before going in the depth of this particular topic, we'll check some syllabus guideline, what all things we need to cover in this particular section. Let me share my screen. So before, this is a periodic channel in which I cover all the Cambridge topics with paper pattern discussion as well as the past papers discussion. So today we will discuss about the chapter 4, and then later series we'll talk about the questions. Right, so let me share my screen here. So this is Cambridge IGCSE Biology 0610 code, and the exams, the syllabus is valid for 23, 24 and 25 exams, June and November series. And we are going to discuss chapter 4. So let me take you to the chapter 4. Yeah, here we are. So, yeah. So chapter 4 is your biological molecule. Will deals with some basic biological molecules such as carbohydrate, fats, protein. Then we will talk about the examples of these biomolecules, such as in carbohydrate category, we'll talk about starch, glycogen, cellulose, and all these molecules form from the glucose molecule, which is a monomer unit. Then proteins forms amino acid, and fats and oil forms fatty acids and glycerol. This is the basics of the biological molecule. Then we will talk about the test, how we can test all these molecules present in the food molecule, present in the food. Then we have the structure of DNA. We'll talk about the two helix structure, which forms double helix. We'll talk about the chemical bases. We'll talk about the base pairing, then in which the pairing will be between A and T and C and G. So as is mentioned in the syllabus that full names are not allowed, so we can write letters A, G, C, T. Then that's all we have in chapter 4. So let's begin. So here we are talking about biological molecule. So as we say biological molecule, this topic is also referred as biomolecule. All biomolecules, like take an example, carbohydrate, protein, fats, lipids, then DNA or RNA. All these are your polymer unit. They are big molecule. They are polymer, and all these are made by a monomer unit. Monomer unit is the single unit. Like take an example, this glucose, right? So glucose is the monomer unit. Monomer means one. When many, I'm putting a number, many glucose molecule will get polymerized and they will form a big molecule which is called polymer. In this case, the name of the polymer is polysaccharide. Polysaccharides. In the same way, there is amino acid, and we have 20 different types of amino acid. When these amino acids are polymerized, we get protein, and fatty acids and glycerol. When these two molecules combined, then it makes one unit, right? This is one monomer unit, and they will form lipids. And in case of DNA and RNA, here I mentioned, there is a monomer unit called nucleotide. Many nucleotides combined to make nucleic acid. Right? I will write it again. Nucleic acid. So nucleic acid is consists of two. You can call either DNA molecule or RNA molecule. We'll talk about nucleotide detail in this topic. So this is just a basics, like what is polymer, what is monomer. Let's begin with the topic. Okay, so we know that all biological systems is made up of the core four biomolecules. Whether it's a plant or animal, insects, birds, virus, you will find any of these biomolecules in the cell. Like in case of virus, you will get protein and DNA or RNA. In normal cells, you'll get carbohydrate, fats, lipid, protein, DNA. So the whole idea of this unit is to understand the biological molecule and how they interact. Right? So in the syllabus, we'll talk about all these four. This is the first one. We'll talk about carbohydrate, second protein, third is lipids, and the fourth we'll talk about DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Fine, so let's continue. Now, when we talk about the constituent, like what all molecules are present in the biomolecules. So in case of carbohydrate, you will find carbon, you will get hydrogen, you will get oxygen, you will get nitrogen. Sorry, there is no nitrogen, there is no phosphorus, and there is no sulfur. So whatever is written in this is just for extra information. It's not given in your syllabus. So carbohydrate example like glucose. So we write C6H12O6. So we are getting carbon here, we are getting hydrogen here, right? We are getting oxygen here. Protein. Protein contain nitrogen. Proteins, they have nitrogen to take care. And some of the proteins also contain sulfur, right? So like take an example of the how protein is represented. Protein is represented as like this, C double bond OH. There is a carbon, then there is a NH2, there is hydrogen, and there is a R variable group. Fats, lipid, again, there is carbon, hydrogen, oxygen. The chemical constituent of carbon, carbohydrate, and fats and lipids are same. There is no change. Now, in case of DNA and RNA, you will get carbon, hydrogen, oxygen, nitrogen, phosphorus. Yes, phosphorus, but there is no nitrogen. Sorry, there is no sulfur. Right. So this is the, you can get a question in your multiple choice from this section. Okay. So this is just an idea. Let's go ahead to the next topic, next slide. Okay, fine. So to understand all these big molecules, we have to understand the monomer and polymer. So in the beginning, I have discussed monomer is the single unit, and polymer means many unit. Means there are so many, like one people, like one person standing in a ground. So we call them one. Suppose there are 10, then you'll say many. And there are so many, then you say it's the field is highly crowded. There are so many people. So you can't call it a 1 or 2 or 10 or 20. There are many people. So in the same way, when many monomer unit combines, you'll get a big molecule, which is called polymer. Example of the polymer is carbohydrate, fats, proteins, lipid, DNA, RNA, and protein. Next, now let's talk about the individual carbohydrate now. So what is carbohydrate? Carbohydrate is also referred as a simple sugar because it's a long chain of glucose molecule, which is also called simple sugar. Why simple? Because after this, you can't get a simplest sugar than glucose. If you are mixing something in water, like suppose you have taken sugar molecule and you are trying to dissolve in water, it will convert into glucose ultimately. After that, there is no simple form of this molecule. That is why we call them simple sugar. Glucose is a simple sugar. When the two glucose molecules will join, we call them disaccharide. Di means saccharide means sugar. So two sugars makes disaccharide. Example of disaccharide is lactose, maltose, sucrose. These are few examples. When many glucose molecules are joined, then they will make a complex polysaccharide, which is called starch, glycogen, and cellulose. Cellulose is the structural carbohydrate, structural carbs, whereas starch and glycogen is the storage carbs. Starch is the storage carbohydrate of plant, and storage reserve food material for animal is glycogen. The main function of the carbohydrate is to provide energy, also to store the food material, also stored in the form of starch or in form of glycogen. And also very important is the cellulose, which is a cell wall component. So the question is, what is the role of carbohydrate in the cell, or what is the purpose of the carbohydrate? You can write these 4 points. And the green points are the important terminology which is must be written in the answers. Let's go to the next slide. Okay, so this is some examples. Monosaccharide, disaccharide, and polysaccharide. Now we come to know there is 3 types because mono is the single unit. When 2 units are there, we call di, and many are there, we call them polysaccharide. So this is many, this is 2, and this is 1 unit. Example, glucose, fructose is a monosaccharide. Many glucose combines to form 2 glucose combines to form maltose. When many glucose are added, they will form starch, cellulose, or glycogen. Next, now we'll talk about proteins. Protein is a long chain of amino acid because it is a polymer unit. So this is polymer and this is monomer unit. And so far we know there's 20 different types of amino acid. And the basic structure of the amino acid is this one. This is hydrogen. This is carboxylic group. This is amino group. And this is the variable group. When we say variable, so this structure is till here. This common in all 20 types. Only R group is different. So R group is different from each one in the amino acid. Right, so this is just an kind of representation. When all the single units are separate, we call them amino acid. When they combined in a long chain, we call them peptide. And when they are present in a very complex form means a complex structure, we call them protein. And proteins have very versatile function in the cell. They act as a structural protein. They also forms enzymes. They forms antibodies, transport system. In a cell, we have a transport channel protein. So they are also made up of protein. Pumps in the cell. They helps in transportation. They can, hemoglobin contain irons, which helps in transportation of the oxygen, the respiratory gases. This is all about your protein. Now, next we'll talk about lipids and fats. Lipid, again, as lipid is a very complex molecule made up of triglycerides. Now, when we see the basic structure of the lipids, the lipid contains first glycerol molecule, second fatty acid, so that is third and fourth. So this is all fatty acid. So if glycerol is connected to 1, we call mono. If it is connected to di, 2, we call di. If it is connected to 3, we call tri. So when we say triglycerides, the meaning is glycerol plus 3 fatty acids, like this. In the diagram, you can see molecular structure. This fatty acid tail have very unique function. The entire structure of the lipid is based on these fatty acids. Fatty acids may be saturated, may be unsaturated. So this is not in the syllabus. We can discuss about this in the later stage in AS and A label. So far, we have to understand what is triglyceride. So when we say triglyceride, the meaning is 1 glycerol molecule connected to 3 fatty acids. And each fatty acid chains are different in shape and size. So it's not fixed how many carbon they should have. So lipids are again divided into fats and oils. So fats, they are solid at room temperature. There is oils, they are liquid at room temperature. That's the difference. So whenever you found any lipid molecules which is solid at room temperature, so they are fat. And when there is the fat, the lipids are in the liquid state, we call them, they are in the oils form. This is all about lipids and fat. Let's talk about the food tasting as per the syllabus. So we have first test, that is reducing sugar test. We also call them Benedict test. So to do the glucose test, we have to take Benedict solution. We have to remember the chemical name. So for glucose testing, we need Benedict solution, and we have to add this solution in a food sample. Then you have to heat this particular solution for 5 minutes. Temperature, you can write 60, 70 or 80 degrees Celsius for 5 minutes. Then you have to see the change in the color. So a positive test will be shown, a color changes from blue to orange or brick color. So like this, the color will change. So in the beginning, it will be blue, then it will be slightly greenish, then orange, and then this red color or brick red color. So when you are getting brick red color, that means it is a positive test. If we are not getting any color from this, that means it is a negative test. So this experimental setup shows how to do the test. Questions may be asked in alternate to practical paper, or in paper 6 or in paper 5 they can ask. So always remember Benedict solution. This is the first marking point. Second marking point to heat, and the third, see the color change. These 3 points will give you the 3 marks in your exams, whether in paper 4 or paper 3 or paper 6 or paper 5. Okay, we'll go to the next test, starch test, which is a non-reducing sugar. This test is done by iodine solution. So we take the food sample and we mix with the iodine solution, and then we'll wait for the positive test. So result is positive when we are getting blue-black appearance. And right, so like suppose the earlier color was orange, then it becomes slightly brownish and then slowly becomes the blue black. That means it is a positive test. If you are adding any disaccharide, like suppose you have taken maltose or you have taken sucrose or you have taken lactose, these all are disaccharide. If you are mixing iodine in this, there is no color change. So always remember, starch test is, iodine test is only applicable for starch, non-reducing sugar. Fine. Let's go to the next test. Protein test. Again, we need biuret reagent solution, and the color of the test will becomes violet or purple like this. Negative test is blue. So if you're getting blue color, that means it is wrong test, or maybe food sample is not having protein. So to give the positive test, the color should be purple or lilac, you can say. Test for lipid and fats. This test is called emulsion test, where we add ethanol in the food sample. So whatever mixture of food sample you will take, that much amount of, like suppose you have taken 3 centimeter cube of the food sample, then you have to add 3 centimeter cube of ethanol also, right? And then you have to see the color change. So if you're getting a cloudy appearance or milky appearance or white emulsion is formed, that means it is a positive test like this. Sometimes we can't see this emulsion, so we have to change the background. So background you can change, either you can put any color for background to see the white emulsion, because white emulsion is transparent. So you have to see by the help of changing some background of the test tube. Next, very important question they ask in paper 6 and paper 5, the DCPIP solution we use for testing vitamin C. Again, we have to take a small amount of food sample in a solution form, and then we have to add DCPIP solution. And the positive test will show the blue color in a dye disappearing. So first we'll get blue color and then slowly the color will go. Color will get disappear. And here in the negative test you'll get blue color. Right? So this is positive test and this is negative test. This is failed test. This is a recap of what we have done so far. So first we have checked the reducing sugar test by using Benedict solution. Initial color of the food sample was blue. Later on it becomes to brick red color. Starch, we use iodine solution. Initial color was yellow brown. It becomes black, blue black. Protein, we take biuret's reagent. Sample was blue initially. Later on it becomes lilac and purple. Fat, we use emulsion. Initially it was colorless and later on it will become white emulsion. Then in case of vitamin C, we use DCPIP. We have to add DCPIP to the food, and the DCPIP will get colorless. Means initially it was blue. After adding, then color will go. Blue color will be not there. Okay, that is a summary of the entire testing. So this is, we have covered the core part of the syllabus. Now we'll go to the extended part. Those students who have opted for core biology, they can do this much. Now for the extended, there is some extra portions of the syllabus. Before, let's try this. Which element is present in protein but not in carbohydrate? So we know that nitrogen is present only in protein but not in carbohydrate. Which elements are present in fats and oil? Carbon, hydrogen, yes, carbohydrates are present, but they are written only. So it's wrong. Carbon, oxygen only. Carbon, oxygen is there, but not only. So this is wrong. Carbon, hydrogen, oxygen, yes. What is the chemical test for starch? So for starch, we use iodine test. Then what the lipid made up of? Lipid made of fatty acids and glycerol. If you're able to answer all these, you can pause the video and then you can check yourself. If you're able to answer this, you are on the right track and answers are given here. You can check it. Let's go to the next section. DNA and RNA. So before going ahead in this particular topic, let me teach you the basics of the molecule. So as I said, this is made up of a monomer unit, which is called nucleotide. Many monomer unit combines to form nucleic acid. So when we say nucleic acid, it may be DNA, maybe RNA, right? So both is possible. When we say DNA, DNA is a double-stranded and RNA is a single strand. This is your double-stranded. This is single strand, SS. Now let's come to this nucleotide. So nucleotide is a monomer unit. Let me write here monomer. And it is consist of 3 subunits. This is very important to know. Nitrogenous base. Then we have pentose sugar. When we say pentose sugar, means the sugar made up of 5 carbon. This is 5 carbon sugar. And this is phosphate. Nitrogenous base is further categorized into 4 types. Actually 5 types: adenine, guanine, cytosine, thymine, and then uracil. Okay. Uracil, this base is only found in RNA, whereas T only in DNA, whereas AGC found in both. Right. So like suppose this is one nucleotide, this another nucleotide, another nucleotide, another nucleotide. They all are making a long chain. When 2 nucleotide chains are joined like this, you see I'm showing a bond between them by hydrogen bond. This is called double-stranded DNA. If a nucleotide is only present in a single long chain form, this is the RNA molecule. We'll go in the detail of the base pairing, how the DNA forms helix. Let's go ahead. So DNA is actually stable in the form of helix like this. This is the helical form. DNA is not stranded, not stable in a straight form. So they are twisted, they are coiled in such a way so that molecules become stable. So structure of DNA molecule. So DNA is again, it's a deoxyribonucleic acid, the full form of the DNA. And it consists of 2 strands of DNA or 2 strands of nucleotide. Right. So what will be the structure of the nucleotide? See here. This is the base. This pentagon is your pentose sugar and this is phosphate. This is nucleotide. This is one nucleotide. So many nucleotides will join and they will make a double helix structure. And then the DNA is quite big. So we have to compile the DNA in the chromosome form like this. And then the information of the DNA is stored in the chromosome, and chromosomes are present in the nucleus of the cell. Okay, so it's getting complicated. We'll talk about the structure soon. Now, so as I said, like one strand, like suppose take an example, this is one nucleotide, this is another nucleotide, this is another nucleotide, this is another nucleotide. There are like this, there are long chain. Let me write the base name. Suppose here you have A, G, C, T, A. This is another nucleotide chain. So one chain combines with another chain by hydrogen bond. I'm just writing H bond. And the hydrogen bond is formed between the bases. So A binds with T, G binds with C, C binds with G, T binds with A binds with T by hydrogen bond. There is a bonding between the strands. So all nucleotide contain the same phosphate and the sugar molecule. The only differ is the base. The base sequence will decide the protein which is going to form in the cell. So always remember the pairing between adenine is always with thymine and the cytosine is always with guanine. And then later on the structure will get twisted like this to form helix structure, helical form. So you can see here in the detailed structure, this is your base, this is your pentose sugar, this is your phosphate. Here also, this is the base, pentose sugar and phosphate. See how they are arranged. If you can observe clearly, you will find that this particular strand, this side, is in opposite direction. So one is going in this way, another is going in another way, right? That is why the orientation and the direction of the strand is opposite. That is why we call them anti-parallel. DNA is anti-parallel in nature because of the difference in the direction. Direction is different, right? Opposite direction due to opposite direction of nucleotide chain. And so chains are different. Base pairing between T binds with A, G binds with C. We call them base pair. And DNA controls everything in the cell. Everything, your skin color, your way of talking, speaking, sleeping, walking, running, anything, whatever you do is a result of the expression of the gene. So DNA controls everything inside you, each and every part. Okay, so we are end to this topic. So before, let me have one question answer session. So you can stop this video and you can solve this question. And meanwhile, I will answer you. What is the name of the basic unit that make up the strand of DNA? Nucleotide. What are the DNA base pairing rules? A binds with T, C binds with G. The two strands of DNA are held together by hydrogen bond. So I hope you all have understood. And we are end of this particular topic. And those who are interested in getting the notes, they can, meanwhile, they can write the notes and they can it. This is the syllabus what we have discussed. Thank you, everyone, and we'll see you in the next video with the enzyme topic. So far, thank you.